Mushroom cultivation. Mushrooms are the fruiting bodies of a fungus, just like apples are the fruiting bodies of an apple tree. A mushroom, or Agaricus bisporus in Latin, is a kind of fungus that are cultivated in at least 60 countries. The oyster mushroom, Pleurotus austriatus, and the shiitake, a Japanese fungus, Lentinula ododes, are two examples of cultivated mushrooms and commonly grown at industrial scale. The mushroom is classified as a heterotrophic creature in the vegetable kingdom, lower plants. These heterotrophs, unlike higher green plants, are incapable of photosynthesis. Fungi are nature's scavengers. Waste goods such as chicken manure, horse manure, straw, gypsum, and wastewater from their own composting are also utilized in mushroom cultivation to provide a high-quality substrate on which the mushrooms can grow. Before it is returned to nature, ammonia is removed from the processed air using ammonia washer. In composting, even ammonia from the air is employed as a source of nitrogen. The fungus, also known as mycelium, uses compost as a source of energy for its combustion, which releases energy for growth. Everybody can conduct this mushroom cultivation. For example, there are websites called agricareer.com that was established by agriculture officers specifically for agri-students and farmers. It is because they spread agricultural knowledge and also sell various of things related to it. So, let's see the methods that were used by them on how making mushroom cultivation.
There are 6 main steps involved in mushroom cultivation. The first step is phase 1 composting. There are 2 types of compost. First, natural compost. Natural compost is made of raw materials like straw, horse manure, poultry manure. Second, synthetic compost. Synthetic compost is made of raw materials like bran, straw, urea, calcium ammonium nitrate or ammonium sulfate, and gypsum. In phase 1 composting, we make compost by mixing and wetting the raw materials. Once the materials are wet, and aerobic fermentation starts as a result of the growth and reproduction of microorganisms. Phase 1 composting takes at least 5 to 18 days for this compost to be ready, and in how many days it will be completely ready depends on the raw materials. The second step is phase 2 composting. The main focus of phase 2 composting is pasteurization and removal of ammonia which formed during phase 1. Concentration higher than 0.7 is dangerous to mycelium span growth, so this is important to remove ammonia. 125 Fahrenheit to 130 Fahrenheit is the most perfect temperature because the ammonia-feeding organisms grow well in this range. At the end of phase 2, the temperature should be at approximately 75 Fahrenheit to 80 Fahrenheit. The nitrogen content should be 2.0 to 2.4% and moisturizer content should be 68 to 72%. The third step is spawning. The process of sowing mushroom mycelium into the prepared beds is generally known as spawning. We can do spawning in 2 ways: by dispersing the compost on the trees or mixing the grain spawn with compost before filling the trees. Once the spawning is done, we cover the trees with newspaper and then sprinkle some water to maintain moisture content. The fourth step is casing. When we mix finely crushed rotten cow dung with garden soil that is called casing soil, and this pH should be on the alkaline side. The casing soil has to be well sterilized and capable to kill the harmful insects, nematodes, pests, and many more. By treating the soil with formalin solution or by steaming, we can sterilize it. The casing soil requires lots of air. After spreading is done, the temperature should be maintained at 25 Celsius for 72 hours. The fifth step is pinning. The pin develops when the carbon dioxide content of the room is less than 0.08%. Mushroom occurring at the beginning develops after rhizomorpha have formed. The newly formed mushrooms are very small. The timing of fresh air to introduce in the room is very important. Prevent the air to enter in the room until the mycelium has begun to show at the surface of the casing. Stop watering the casing when the pin initial starts forming. The last step is harvesting. We rip the mushroom into flashes. The first flash is ripped in 3 to 5 days and get the yield of 15 to 20 kilograms. The second flash is ripped in 5 to 7 days and get the yield of 9 to 11 kilograms. A little less than first flash. We get about 27 to 35 kg total yield. Hand-picking mushroom can be stored and consumed fresh.
Mushroom cultivation is a technique for cultivating mushroom from waste from plants, animals, and industry. Mushroom cultivation is an indoor activity. There are problems resolving mushroom cultivation. Firstly, such as mycelium fails to form. This is probably because improper initiation strategy, chlorinated or contaminated water, bad pasteurization, or bad substrate. Secondly, mycelium grows but fails to produce mushroom. The cause or substrate formula is not suitable, pasteurization was too quick, too high density in the incubation area, there are not enough ventilation to decrease accumulate temperature, or the hygiene of the incubation house. Next, the technology that enhance or ease mushroom cultivation, such as computerized control, automated mushroom harvesting, compost preparation, mushroom cultivation on non-compost substrates, and innovative methods of substrate sterilizing and spawn preparation will all help to boost mushroom culture productivity.
In a nutshell, many people can practice mushroom cultivation as its methods are easy, as long as you have the materials and know how it works. Even though it looks easy, we must trust its physical and biological functions. For example, using a big room more than 1500 m3 in width for mushroom production with absolute humidity and out of direct sunlight. These are good for mushroom growth and will produce healthy and quality mushrooms. Besides, make sure that the room temperature is around 13 degrees Celsius to 16 degrees Celsius. The range of this temperature not only favors mushroom growth, but cooler temperatures also can lengthen the life cycle of both disease from pathogens and insect pests. Lastly, water input is crucial because too much will restrict oxygen supply by occupying pore space, while too little would limit bacteria and fungal growth.